is Rocky Cranenburgh, and I'm the CSO of Prokarium, which is a relatively new biotech company in the UK. And I'm going to talk to you today about our vaccine delivery and production platform, Vaxonella, which uses live attenuated Salmonella to deliver vaccines orally via the lining of the small intestine. So we're a spin out from a contract manufacturing company I used to work for called Cobra Biologics. Cobra manufacture proteins, DNA, viruses, even bacteriophages and bacterial cells for clinical trials, those experimental medicines. And we're located in Keele in Staffordshire, so midway between Birmingham and Manchester in the northwest. And that's our site and our platform. So what I'm going to do today is mainly just give a brief overview of the whole platform using Salmonella and a little bit about the vaccines we're developing using that. So our focus is to re-engineer Salmonella to turn it into what is a very serious pathogen. Our human delivery system is Salmonella Typhi. So the species is Salmonella enterica. And this includes a range of serovars, including Typhi, which is our mouse model and has a very wide species range, pretty much anything with 4 legs or 2 legs, down to Salmonella Typhi, which is our human delivery system, which exclusively infects humans. It doesn't have any other reservoirs in the environment. It doesn't even infect the great apes. So to turn it into a suitable delivery system that's safe to use for vaccines and immunogenic, we have a number of changes to it, and one of the key events for our company is the acquisition of the Typhoid vaccine development program from Emergent Biosolutions in the States. So they'd been attenuating Salmonella, and our chassis strain, as is the term in synthetic biology, is a strain called ZH9, which is an experimental typhoid vaccine that we've now acquired, and we're re-engineering it to turn it into a delivery platform. So it's got 2 main mutations, one in aroC, which encodes chrosimate synthase, which is involved in aromatic amino acid biosynthesis, and to replicate it needs 2 aromatic compounds which we supply in the growth medium. When it's growing, it grows very happily. You can handle it exactly like E. coli, so it's a very easy strain to grow to high densities and to genetically manipulate. Also, it has a mutation in the SPI2 locus over here in a gene called ssaV, and this is part of Salmonella's type 3 secretion system, which it uses to inject effector proteins into the cell to help its colonization of the gut epithelia, and then its later dissemination via the blood system and the spleen. So these are the 2 attenuating mutations, and we also have other genetic systems that I'll explain in more detail, which allow us to replicate plasmids, because we've got multi-copy plasmids where our recombinant vaccine genes are replicated, and we don't use antibiotic resistance genes, because there was a talk yesterday that mentioned the fact that regulators are trying to eliminate antibiotic resistance genes from various biotherapeutic, including vaccine applications. They're already not allowed if you're using a live bacterial vector, so we can't use antibiotic resistance genes anyway. So I'll talk to you about the other systems we use. One is for the automatic deletion of the antibiotic resistance gene, and then the replication of those plasmids once that gene's deleted. So the principle of vaccine delivery using Salmonella is as illustrated here. So whether you have a human and you're using Salmonella Typhi, or a mouse and using Salmonella Typhi, it's delivered by obviously orally. So we eventually want to develop a pill that we can use, but at the moment it will be a suspension that is drunk with bicarbonate buffer. Mice are vaccinated by having an oral gavage, so a blunt needle that delivers the bacterial vaccine directly to the stomach. So the Salmonella then invade via the microfold or M cells in the lining of the small intestine into these lymphatic nodules called Peyer's patches. And then in Peyer's patches, they will either invade or be phagocytosed by antigen-presenting cells. So this is their natural method of infection. And then they can alter the phagosome for quite a while, so they're not immediately digested, but in there we use a promoter called ssaG, which is only induced at the point of phagocytosis. And that means that the bacteria are not burdened by continually expressing the recombinant vaccine that we've engineered them with, but they only express it exactly when needed at the point of antigen presentation. And this is very good at eliciting both MHC class 1 and class 2 responses. It tends to be that the responses that we measure are related to the particular pathogen that we're working on, the particular disease target, and whether those are required. But we do see both branches of the immune system stimulated. They'll eventually be lysed by fusion with the lysosome, and then you get further antigen presentation that way. So that's the basic delivery system, which is exactly the same in mice with Typhimurium and humans with Typhi. So this is just an overview of the system that we use to maintain the plasmids. It's just a cover picture if anybody wants to print these sort of things out. But before I go through it, it's just you're not allowed antibiotics with these systems, and there are lots of ways of getting around that. And the typical ways are to take a essential gene on the host chromosome and mutate it and then put the functional copy back into the plasmid. So that's a complementation approach. Or there are the post-segregational killing mechanisms such as HOC-SOC and the CCDA, CCDB systems, where you have a toxin-antitoxin, antidotes on the plasmid's lost, the cell's killed. The problem with all of these systems is that they share the same disadvantage as the antibiotic resistance genes in that you're expressing a second or an additional recombinant protein which is not needed, and it's the expression of that extra recombinant protein that often leads to plasmid loss. So if you take E. coli as an example, you're very used to having an antibiotic resistance gene, antibiotics in the medium, and of course the antibiotics are to prevent cells that have lost the plasmid proliferating. But the thing that's most likely to cause them to lose the plasmid is the antibiotic resistance gene in the first place and its constitutive expression. So it's a bit of a chicken and egg situation. So we thought that the best approach would be to completely eliminate any unwanted expressed genes. So we have an approach called operator repressor titration, or ORT, and when we apply this, it was originally and it's still being used in E. coli for the production of DNA vaccines and recombinant proteins. We moved it into Salmonella and called it ORT-VAC, but the principle is the same. So to illustrate how this works, we've taken an essential chromosomal gene called DAPD, and this encodes an enzyme which is produces the amino acid DAP. And DAP cross-links peptidoglycan in the bacterial cell wall, so it's absolutely essential, and without it the cell would lyse. And then what we've done is we've replaced its natural constitutive promoter with the TET operator and repressor promoter. So we now have a system where the essential DAPD gene is controlled by this regulated promoter. So with a sublethal concentration of tetracycline, we can add that to the medium, and that will switch on expression. So this is what we'd use to grow it. Doesn't need to be delivered to any of the mice or humans. So it's only for culturing. At the very initial stage. It's not even required in a larger scale culture. Then, when the operator sequence that's on here, which is just a short non-expressed sequence, is also found on the multi-copy plasmids that we use. So normally, you'd have the repressor, which would be produced here and bind to the operator and would shut it down. So that would shut down expression. But also, another way of de-repressing this, instead of adding the inducer, is to have the same operator sequence present on a multi-copy plasmid. So here, the repressor proteins, because the plasmid's present at a much higher copy, will preferentially bind to the plasmids, and you get expression of the essential gene and cell growth. And then also, the only gene that you need on here is the antigen gene of interest, which is then expressed. So that's really the system in a nutshell. So we can select directly using this system. So you can you can cut out the antibiotic resistance gene, religate and transform into these strains. But, of course, we do all our cloning in E. coli. We want to put them into Salmonella. There is actually a restriction barrier between the two species, which means that we get a massively reduced transformation efficiency. And we just want a really simple system that's easy to use. To get around that, we invented an extra technology that goes along with ORT-VAC called XMark. And to explain the background to this technology, you need to know about the XER recombination system, which is naturally present in a lot of bacteria with circular replicons, and in E. coli and Salmonella, which are of course very closely related, it operates in this way. So plasmids present in multi-copies per cell are, of course, identical. So rec-A, which causes homologous recombination, and it's essential for repairing double-stranded breaks in chromosomes, will recognize that there are two plasmids here and recombine them into a dimer. So that's normally a problem, especially in E. coli strains where you might want to, say, make a recombinant protein there, because the dimer then becomes much bigger and bigger, and it gets so big, then something called the dimer catastrophe occurs, and the plasmid is lost from the cell because it can't maintain it, can't replicate it quick enough. So to avoid this happening in nature, the XCR system has a recombinase called XCRC and XCRD. These two proteins collaborate to recognize that there are these two copies. And it acts on sites called Psi, which are derived from Salmonella, or a very closely related site called SIR from E. coli, and which has accessory sequences as well. The accessory sequences being recognized by these two accessory proteins. So when the dimer has occurred, the XCR system recombines them back together and you get monomers again. So it prevents this accumulation of dimers leading to dimer catastrophe. But the upshot of all this is that if you take a sequence and put it between two copies of the Psi sites with its accessory sequence, it can effectively be deleted. So this is the same mechanism as the Cre-Lock system, for example, and the lambda recombinases. So what we did was essentially use that system and build plasmids which had an antibiotic resistance gene. We've used chloramphenicol, which has these, which is flanked by these sites. So you've got the Psi site here, either side. This is a typical plasmid for protein expression, where we also have the ssaG promoter that drives expression of a clone gene, which would be here. All we need is the short, 20 base pair non-expressed operator sequences for selection. So no additional extraneous genes. And the parloc is useful for helping partitioning between divided cells. So then when we take this plasmid and replicate it in an E. coli PEP-A mutant, that's a mutant in one of those accessory proteins, it's perfectly viable, and it can divide its own chromosome very happily because the accessory proteins aren't needed for XCR recombination on chromosomes, so there's no problem there. They're only needed for XCR recombination on plasmids. So this plasmid replicates quite happily, but then we transform it by antibiotic selection into any enteric bacterium, E. coli, Salmonella, whatever, and then the XCR recombinases recognize these two copies of the multimer resolution site, the Psi site, and will generate both a mini circle over here and a mini plasmid. Now, this mini circle just has the antibiotic resistance gene, and because it has no origin of replication, it can't replicate, so it's eventually lost as cell division takes place. But the proper plasmid has the origin. So this is the one that would contain the antigen gene. So that's the way the system works. And we find that these plasmids are actually so stable that in our candidate vaccine that we're taking through at the moment, I'll tell you about in a minute, the replication of this doesn't require any plasmid maintenance system. So you're firing it in using antibiotic selection, the gene's automatically being deleted, and then the plasmid, because it doesn't have an express selectable marker gene, is so stable that it doesn't actually even need a maintenance system. You can do this in just a bog standard strain of E. coli you can get from a catalogue. Once you've got the XMark system operating. So this is an overview of the whole Vaxonella platform for vaccine development. So what we have is a very quick and very low-cost system of both identifying and testing vaccines and getting them into clinical trials, which is, of course, where you want them to be. So what we start with is we have these two strains with exactly the same mutations. One of Typhi murium for testing in mice. Again, as I said, it's got the same pathology in mice as Typhi has in humans. So we get our gene synthesized, cloned into our plasmids. The whole manufacturing process is exactly the same regardless of what vaccine. If we're making a recombinant protein, you need to start from scratch and have a brand new purification and production system every time for each new protein. Here we don't. We're just making a bacterium, so everything is the same apart from a couple of specific assays for the new antigen that you're trying to produce. We then tested in vitro. We have media that basically have the same conditions as the macrophage, so as the phagosome in the macrophage, so we can look for expression that way and make sure the proteins are being expressed successfully. And then we do mouse experiments, essentially. So we just look for the immunology in mice. If there's a challenge or protection that we can do, then we'll do that as well. And then, assuming everything's successful, the same plasmid is then just moved into our Typhi strain for use in humans. So at the moment, our sister company Cobra is making our lead candidate, a typhoid bacterial diarrhea vaccine, by GMP, just using shake flasks. Basically, with 4 1-litre shake flasks, we'll get enough material for clinical trials, because all we need to do is essentially grow them, centrifuge them, wash them, and make glycerol stocks. And what the volunteers at the trial centre will have is basically just the usual 20% glycerol that you're familiar with, thawed out, dunked into buffer, and drunk. So it completely eliminates all the very costly and time-consuming downstream purification that's associated with the development of experimental vaccines, and which I know all too well from my many years at Cobra on the other side of the fence, trying to make these for other companies. So you can then do toxicology, which has to be done, but in our system is basically meaningless, and then a phase 1 trial in humans, which is what we're heading to at the moment. So that's an overview of how you can easily slot in new antigen candidates for testing in the Vaxonella platform. So our first candidate is a dual vaccine against typhoid and bacterial diarrhea due to ETEC, which is Enterotoxigenic E. coli. So in our particular case, in the first instance, the bacterium itself is the typhoid vaccine component, because it's got epitopes like the LPS and the VI antigen, which are essentially a typhoid vaccine. For future vaccines, that won't be a focus. But the second, the actual recombinant part, is from ETEC, and this is probably the most common cause of bacterial diarrhea. It's probably either equivalent to or second only to rotavirus as the main cause of travellers' diarrhea. So about 1 in 6 of travellers from the developing countries will develop will contract ETEC, and it's a very unpleasant form of diarrhea. And it's not just a problem there; it kills up to 500,000 children every year. And there's no dedicated vaccine available. So this shows, this is data from a PATH BVGH report that shows that you get about 10 million travellers per year infected by ETEC, which is about 45% of those travellers' diarrhea cases. And typhoid, although there are multiple vaccines available, is still a problem, and those organisations seeking to develop next generation vaccines, which will actually be more efficacious at addressing typhoid as well. So here we'll have a combination travellers vaccine. So some very early data. I can't show you all the latest data, unfortunately, due to confidentiality reasons, but just to let you know the overall plan of the vaccine design. ETEC produces 2 toxins which cause the diarrhea, the labile toxin LT and the stable toxin ST, and ETEC is basically defined by either one or the other or both of those toxins. So all ETEC strains will express LT and or ST. And also we've included a single recombinant protein fusion which has epitopes from these 2 and also from multiple colonization factors, and these are the fimbriae, the actual physical structures that help with the bacteria to adhere to the gut epithelia. So we have vaccines candidates with combinations of these delivered by our Salmonella, and this just shows an earlier experiment where mice were dosed with both the empty vector, so this is just the Salmonella on its own. Blue is the LPS. Then you've got the LT, and these are antibodies, IgG, against the recombinant vaccine. And ST is grey, and one of the colonization factors is shown in yellow here. So we get both. So the sort of typhoid proxy is the LPS response here. So in our recombinant strain, we get the levels that are about equivalent to what would be protective for typhoid from ETEC in terms of the immune responses against those. The IgA results basically look identical to the IgG, extremely similar. I haven't showed those there. So that's basically very similar to the final vaccine that we're taking through. We've modified ST, so it's less likely to cross-react to native human hormones, guanylin and uroguanylin. So along with this, and as the next stage of development, again, as I said, we wanted a capsule to be able to be the final delivery system. So we worked with Cambridge University to develop a formulation called BAR, which is a bile-adsorbing resin. So, and the reason for this is that we found that when you dry Salmonella and then have an enteric coating, that's fine for getting them through the hydrochloric acid in the stomach, but then they need to be released into the small intestine and they'll be released straight into bile acid, and this causes a multiple order of magnitude reduction in their viability. So by encapsulating the Salmonella with the bile-adsorbing resin, which can be a range of compounds including cholestyramine, the cholesterol drug or treatment, you basically absorb all the bile acids to the surface of the capsule, and water is allowed to enter freely. And after only a minute or two is sufficient for the bacteria to rehydrate and gain their natural bile resistance. And then the capsule will break up and the bacteria are released. So if you have a look over here, the blue is the rehydration of Salmonella in just a buffer, so just PBS, and the sort of greeny one is in bile. So when you have no capsule and no bar present, you get a multiple order of magnitude reduction in the viability. The capsule does give partial protection even without bar, but you're still losing 1 or 2 orders of magnitude, which can be very significant for a vaccine. But when we have the bar present as well, then there's no detectable reduction in viability at all when delivering dried bacteria into a physiologically relevant concentration of 4% bile. So that would be our eventual capsular formulation for adults, with infants being given a sachet with a buffer. So when our collaborators at Cambridge did a press release on this, it gained a lot of interest. So we think there's a real market out there for an anti-diarrhea vaccine, or Delhi Belly as they've called it. I think it's Montezuma's revenge in the States. And the professor we collaborated with was constantly getting phoned asking when it would be on the market, and had to tell disappointed people it will be ready in about 8 years. So that was the press, and this is our typhoid vaccine when it was. So we inherited it. Well, we acquired it from Emergent, and this is a challenge experiment. So the BBC were reporting on volunteers for clinical trials, and this guy is about to drink 10,000 live typhoid bacteria and then be released onto the streets of Oxford. So this was done by Andy Pollard at Oxford University, and in fact, in an earlier trial, he contracted malaria as well. So maybe this morning's speaker was responsible for that. This will be the sort of challenge that we'd want to do later on when we're testing in phase 2, both for ETEC and typhoid. So the overview is that essentially we've got a typhoid vaccine that's completed all of its phase 1 and phase 2 trials. So it's been tested in 8 clinical trials now on over nearly 400 volunteers, including 100 children. So we know that our Salmonella strain, the Typhi strain, is absolutely safe and produces a very good immune response. And of course, another advantage of using Salmonella as a delivery system is that it's very immunogenic. Obviously, it has LPS so we don't need any adjuvant to go with it. We are working on the combination, so we're taking it right back to the start essentially, and putting the ETEC vaccine, and have put the ETEC vaccine in, and that will go into a clinical trial end of next year. We're also working on Clostridium difficile, which is a major nosocomial infection with a big pharma company, and next in our pipeline will be Chlamydia trachomatis. So another advantage of these systems is that they induce very good levels of mucosal immunity, and that's particularly applicable to those pathogens that invade via mucosal surfaces, which of course a number of them do. Just to summarise the advantages of the system, so of course it's oral, so you don't need any needles. You could eventually be able to pick this up at pharmacies or post it to areas where you need vaccination programmes. There's no adjuvant at all because these are really very strongly immunostimulatory. We've done previous studies on other strains using our genetic technologies where we've got protection against bubonic plague, anthrax and TB. It can also be used to deliver DNA vaccines as well, which I haven't mentioned so far. We get a very broad immune response, both IgG and IgA and T-cell responses. A very nice mouse model between Typhi murium and Typhi with the 2 strains in their cognate hosts. A very safe vector. I mean, the really nice thing about this compared to, for example, TY21A, which is the licensed Johnson & Johnson / Crucell vaccine is that it is really viable. It grows extremely well, very easy to modify, but it's very attenuated, very safe. And there's no downstream purification of protein, so this can really slash the cost of development of new vaccines in a simple manufacturing process. And of course, we're very keen to collaborate with anybody who wants to evaluate the platform for their own vaccine delivery, especially for obviously for recombinant proteins, but we have also got some good evidence of DNA vaccine delivery. I haven't presented DNA vaccine data here. It was nearly as good as the protein vaccine delivery, actually, in this particular case for our ETEC vaccine. So it just remains for me to thank my team at Prokarium, so Paolo Salerno, Annalise Soulier, and our CEO, Ted Fjellman, collaborators at Cobra Biologics, where the vaccine is currently being made. Dave Sherrod at Oxford University invented the ORT system. Nigel Slater and Al Edwards worked on the bile-adsorbing resin, and the mouse experiments that are ongoing at the moment, and you've seen early data from Ian Henderson and Tim Wells at Birmingham University. And big thanks to a lot of people at Emergent for the over many years developing the very safe and immunogenic ZH9 strain and its Typhi murium equivalent, and for funding by the TSB in the UK and BBSRC. So thanks very much. Question over there.
I was just wondering that, presumably when your salmonella gets into the macrophage, it's in the phagosome. Is the antigen being translocated and the bacteria staying within the phagosomes? So how do you get a good CD8 T cell response?
We do, yes. We think they're mostly remaining inside the phagosome. We're secreting the recombinant proteins, and that's probably what helps.
So the secretion is the one which is driving, and there's no antigen translocation into the cytosol, presumably.
Yes, that's right. Yeah. So but we are, as I said, we also deliver DNA vaccines using it. We don't really understand how that happens. But it tends to be more variable with DNA vaccines. At least with recombinant proteins, we always know we're going to get a good immune response.
The second question I had is that, in animal models like mouse models, normally even the mutated, say, aroA strain tends to be quite chronic in terms of bacterial clearance. So what's the kind of profile for the persistence of the bacteria in your animal models?
It's very nicely cleared, both in animals and humans. So you're right with aroA. But with the dual mutations of aroC and ssaV, it is really cleared in a couple of days from the gut. So we don't see persistent shedding, which makes it very safe. And also the mice tolerate it really well. We've tried to use other just aro mutants, and it often makes the mice sick, whereas this is extremely well tolerated. And the dose of 10 to the 10 is the same in mice as we use in humans, 10 to the 10 CFU.
So this is the opposite of that question, which is once you have immunity to the vector, can you keep using it to deliver other vaccines, because presumably you're actually going to neutralise the vector?
Yes, you can. There's a review that actually looked into this. I usually put it up as an appendix slide, but we think it will help it with a boosting effect for that, because you're not actually stimulating the immune response until you get into the phagosome. So it gets through the gut, and then it's phagocytosed, and that's where the antigen expression starts. So there's not that much scope for it to be neutralised before then. So we think that the multiple doses in the future will help with the boosting effect.
That answer makes no sense, because if you're making an IgA response that's really great against your vaccine,
yeah,
you're going to neutralize it before it can get there. You're going to lose the ability to actually use it again. Unless you're not making a very good IgA response locally, there's no way you can use that vector again, or not in certainly the same performance ways.
It depends on the level of IgA response to the Salmonella as opposed to the IgG response, I think. So the recombinant antigen is going to be is obviously going to be different in each stage. So we want the immune response to be sort of reasonable immune response to be generated, but then it's going to be targeted against a specific recombinant vaccine that we express the next time. So we think it actually is going to be beneficial.
There actually is a combined cholera and ETEC vaccine. It's orally available. It's called Dukoral in my country, and it has a good effect against cholera, but the effect on tourist diarrhea is pretty bad. So I wonder, why do you think your vaccine would be better?
Yeah, so Dukoral is licensed in some countries for ETEC. The ETEC protection is due solely to the cholera toxin, which is very similar to the LT component of ETEC. But that's so it's only partially protective. Whereas in our vaccine, we've included ST as well as LT and the multiple colonization factor epitopes as well. So we'll get a very broad, we theoretically will target all ETEC strains, whereas Dukoral has only partial protection against some of the strains. So on the pie chart I showed, you're losing 2/3 of the strains that Dukoral couldn't protect against because they're also producing ST. So we're looking for toxin neutralizing antibodies here.
Actually, just a quick question on, is there size limitation for like a transgene delivered product in your plasmid system? So, I mean, if you wanted to now move to a second target, like you're saying, for another diarrheal disease, do you have to now have another Salmonella strain delivering, that transgene as part of a plasmid, or can you kind of combo it up and have it in the same Salmonella cell?
Well, we can put genes of multiple KB in there. So our C. diff vaccine is several KB genes. So if we wanted to put in another vaccine, we could probably do it in tandem, or we'd use 2 strains. So it depends on the amount of DNA we're putting in. But it can certainly replicate plasmids or inserts that are several KB.